Hello everyone, welcome to our grade 9 biology class. Okay, so I'm very happy today that I can take you by online class. Okay, so we will study the new lesson today, but let me review some question in the last lesson. Okay, now what is pathogen? Okay, try to answer by your own self. Okay, number 2, how many types of pathogen are there? What are they? Okay, number 3, do you know the name of disease that can kill many people worldwide today? Okay, so let we check the answer all together. Yes, the first one is pathogen are microorganisms that can cause disease, like we studied in the last lesson. Okay, so how many types of pathogen are there? There are 4 types of pathogen. They can be virus, bacteria, protists, and fungi. And I am sure that you know this and you can answer this clearly, right? What is the name of disease? Yes, it's corona disease. Okay, and so what kind of pathogen that cause corona disease? What is its name? You know or not? Okay, perhaps I know you know this clearly also. Yes, it's a kind of virus. Okay, among the 4 pathogens is virus, and its name is COVID-19. Okay, so now move to another slide, please. Okay, number 5 is the last review question. What are the diseases that caused by the 4 types of pathogen? Bacteria, protein, fungi, and virus. Okay, so because we study already this, okay, let me check this in this table. Okay, there are 4 agents or 4 pathogens, right? Okay, there are many diseases that caused by bacteria. There are many diseases that caused by proteins. Okay, many diseases that caused by fungi. It can be 2, but it's more than this. Okay, and it can be many diseases that caused by virus. Okay, like today we have cold, influenza, AIDS, measles, mumps, polio, smallpox, and also like today is COVID-19, right? It's a virus that causes pneumonia, like okay, chronic pneumonia that kill many people worldwide. Okay, so okay, so today we start a new lesson. Okay, in the section 3, and in the chapter 3 the same like the last lesson. Section 3, infectious disease. Okay, before we study, we need to know our objective of this lesson is you can describe the disease in history, and you can define word pasteurization and Koch rule, and you also can explain the step of Koch rule. Okay, so let me move to another slide about disease in history. Okay, so I want you to observe the table at the right hand here. You can see it's a table that shows you about the death of the epidemic influenza in 1918, right? Okay, so there are many people around the world. Okay, in many countries like United States, United Kingdom, India, Africa, Australia, and many people are killed because of the influenza. But not only the influenza, it can be plague and smallpox also can kill million people worldwide. And it estimated that during 1918 influenza outbreak, 20 million to 40 million people die. Okay, today the cause of the disease can prevent or cure them. But even today there are diseases such as AIDS or corona or Ebola also, yeah, it can not be cured, right? Okay, now move to another slide, please. Talk about the discovering disease organism. Okay, you can like you see the picture here is show you about the first microscope that invented by Van Leeuwenhoek. Yeah, he invent his first microscope is like that. But you see this one is the microscope that we use today in our lab. Okay, by using the microscope, you can observe many small organism like microorganism like mold, spore, yeast, or bacteria, right? And in the 17th century, by using the microscope, okay, we can observe this organism for the first time. But it took about almost 200 more year more to discover the relationship between some of the microorganism and the disease. And scientists did not make a connection between virus and disease transmission until the late 18th and early 19th. Okay, so you know who? Who is he? Okay, I think you know him. Some people know him. He is the famous scientist. His name is Louis Pasteur. He's a French chemist and microbiology. It means he also chemistry. He studied chemistry and he studied about microorganism. Okay, at that time he learned that microorganism could cause many disease to human, because the microorganism could spoil wine and milk. So he realized that the microorganism could attack the human body in the same way. But at that time many scientists did not believe him. Okay, and another way he invented a process called pasteurization. This process is a process of heating the liquid to a specific temperature that can kill most bacteria. Okay, we also use this process, like today we use one saran, right? For when we inject vaccine or medicine to the patient, we use only one saran and we throw away. But before, one saran can use many times. So how can we use it for many times? So one saran need to be killed or sterilized, right? By using the heat of liquid, or we can boil it, or yes, another way. So we also use the pasteurization. Okay, so now look at another scientist. Here is the Robert Koch. He is a German doctor. Okay, so because at that time, many scientists did not believe Pasteur, because Pasteur don't know how to tell which specific organism cause which disease. He only say yes, microorganism could cause disease, but he didn't know how to tell which bacteria, which is bacteria, is virus, is protein that cause the disease. So he is not sure, he don't know. Okay, that's why the other scientists did not believe him. Until the late 18th, there has a young German doctor. His name is Robert Koch. He developed a series of methods for identifying which organism was the cause of the particular disease. This process is called Koch rule or Koch postulated. Okay, and this process is still used today. So let's move to the process. Okay, here this is a method, and I can ask you how what is Koch rule. Okay, we can answer it's a series of methods for identifying the organism what the cause of a particular disease. Like today, before we want to know is we can know COVID-19 is the pathogen that is a virus that cause the disease today. So scientists need to follow by this step. So look at the first step. How many steps are there? Yeah, there are 5 steps. Okay, but in first step, second step we can divide into 2 A, B, third step, fourth step, and fifth step we can divide into 2 A, B also. So now let's move to our first step. Look at this, the rat or mouse. It died, right? So it died, perhaps it have a microorganism that cause the disease, right? That cause it die. So this animal, the dead animal here now, it contains many microorganism or bacteria, or we don't know what kind of pathogen is. It can be virus, bacteria, protein, or fungi. Now example, if one organism die like that, okay, we isolate, scientists isolate the microorganism from this dead organism here, the dead animal here, and isolate it from the blood, from the poop, etc. And then we move it and grow it in a pure culture here. Now microorganism, we don't know what kind of it. Okay, and move to B here. That's the second step. The microorganism now, after we culture it, we grow it, so we can observe it alone, right? Only the microorganism, not the other particle from beside it. So a microorganism now is observed under microscope. It can be bacteria or virus, okay, and then after we the scientists observe it already, they take a photo or they draw the shape of it, okay, and then now this microorganism is identified already, right? And then we inject this microorganism to another healthy animal, okay, another healthy animal. Normally we always use the rat or the mouse because it's a mammal like us, okay, like human. So we take it and it means like now microorganism here is inject into the healthy animal here, and then after that what happened in such step? It die, right? Yeah, it die again. Okay, so the disease is produced in the second animal, and microorganism is isolated from the animal, okay. After it die, this animal die, it become the microorganism now isolate again, and it will grow in a pure culture again, okay. And then in this step, after they isolate it and grow in a pure culture, they observe it under microscope again. If this one after they take the photo or draw it the shape already, if this one is similar to the one that they take from the first animal here, it's the same, it means this one scientist can identify that this organism, this microorganism, this bacteria or virus is the cause of this particular disease, okay. Here are the 5 steps of the Koch rule, or we can say okay, it's a step that we can identify which organism was the cause of a particular disease. Thank you for your pay attention. Okay, see you next time.